right, video number 2, so focusing on blood pressure. So earlier in AP 1, where we were looking at gradients, and one of the examples was blood going down its pressure gradient, and so we have to look at that and maintain good levels for our nutrient exchange, and then basically our blood pressure almost pretty much drops off once we're in the veins, but that's okay. So with our blood pressure, that is the force that the blood exerts against the vessel wall. So if my hand here, the right one, is our blood vessel, then it's the pressure of the blood pushing up against that blood vessel wall. So in order for us to measure our blood pressure, we can kind of estimate it using a sphygmomanometer, also known as a blood pressure cuff. So that one is looking at the pressure in your brachial artery. So that's where it's on the arm. They do have some that fit the wrist area, just makes it easier, particularly for older people or some people who have larger arm sizes. Anyway, so the two pressures that we're looking at using a blood pressure cuff are the systolic and diastolic pressure. So again, we don't care what the atria are doing, we're focusing on the ventricles when we deal with systole and diastole. So your systolic pressure is your peak pressure. So this is when your ventricles are contracting. So this is the largest amount of pressure that's going through the blood vessel because your ventricles are pumping out blood into those blood vessels. Diastolic pressure is going to be your lower number, and that is when your ventricles are relaxed, and so there's less pressure because there's just not that physical force pushing it. So our normal values, and I think it's actually even lower than this now, is 120 over 75. It used to be 120 over 80. Yeah, I know. So the 120 is our systolic, and then the 75 is our diastolic. So one measurement that we can look at is the pulse pressure, and this is the difference between our systolic and diastolic, and you don't want them too widely different from each other because your body's having to respond from these big fluctuations. But you do have to have some pressure, enough in order to, again, get your blood to wherever you're going and have enough blood pressure to push those fluid out of the capillaries and into your tissue space. There's also mean arterial pressure. You don't have to know it for the test, but you're looking at this difference between your pressures over time, so not just a one-time measurement, looking at kind of more of a function thing over a longer period of time. Okay, so with your blood, again, because you have your two different pressures, your blood flow in your arteries is pulsatile. So, sorry, this is gross, but if you ever looked at a horror movie or some kind of action film where the person gets injured and the blood spewing out in bursts, so every time you get that psh, out of blood, and that would be when your ventricles are contracting. So that's why you get that kind of burst out of it. Capillaries and veins is going to be a steady because there isn't much of a difference between your systolic and diastolic there. So it's a lot smoother flow. So with that blood pressure, of course, you want to maintain good levels, even though they tend to rise with age because of, well, diet, and eventually stuff gets stuck up in your blood vessels. And then, of course, if your tissues aren't functioning the way they should, so you could end up with arteriosclerosis. So this is a stiffening of the arteries, so therefore they're less elastic and able to respond to varying degrees of pressure. Or the other one is atherosclerosis, and this is where you have a crappy diet, and so you end up with a bunch of lipids being inside your blood vessel, and those start to close up and might possibly give you a heart attack or a stroke. So basically a stroke is just a heart attack, but for the brain. But still same thing, lack of blood flow to that organ. All right, let's say you're doing your blood pressure and it's above 140 over 90. So again, 140, the larger number, is systolic. Diastolic is the lower number. Consequences. You can weaken the artery. So again, it's that pressure smacking up against that blood vessel wall, and eventually it gives up, essentially. Can cause aneurysm. So blood vessel, this is where there is a weakened spot in that blood vessel. And can also promote atherosclerosis. So remembering from the previous slide, your atherosclerosis is where you have the buildup of deposits. So with that weakening and damaging of our blood vessel, stuff can then start sticking to our blood vessel, and that just stuff starts adding up and closing off your blood vessel flow. Opposite situation, hypotension. So hyper meaning a lot, hypo meaning low. So hypotension, this is a low resting blood pressure. So this is caused by blood loss. So not enough fluid inside that blood vessel wall to have that pressure. Dehydration, so that's going to decrease your blood volume, so therefore decrease your blood pressure. And then last one, someone who is anemic, so they don't have enough blood cells. Translation, less blood volume, less blood pressure. Okay, so looking at our pressures as we're moving through our blood vessels. So here's the arteries going away from the heart. So we have this wavy pattern because you have a difference between systolic and our lower diastolic. So again, the big push from the ventricle muscles as you're going out the heart, you have your higher number, and then when they relax, you get your lower number. So over your distance, again, blood flows down its pressure gradient. So when you get here in your capillaries and doing the capillary vessels and capillary dynamics calculations, remembering your dip in your blood pressure and therefore causing your switch in your ability of whether or not you're dropping off nutrients and picking up waste products. Okay, so this is where, again, those little sphincter muscles around your smaller arteries right before you get to your capillaries. So that's going to help control all this waviness. So that's going to help smooth it out, and then you have less effect of your differences, and so you just have a nice smooth decrease in blood pressure. So again, that helps with moving it. But then you get way down here and you get these like bumps, increases of blood pressure. And the reason for that is because you get into these such larger veins where you're starting to collect a lot of blood in those larger veins. And so that's going to cause an increase in blood pressure because you have more volume in those areas. So just a big pile of blood coming into one area. All right, now time to focus on the capillary. So dealing with our exchange with our capillary beds and what happens when that does not go right. So capillary exchange, again, it's our movement of fluid across our capillary wall. So this is where we're able to drop off our nutrients, water, oxygen, glucose, amino acids, lipids, minerals, antibodies, hormones to the tissue, and pick up waste, carbon dioxide, and ammonia from the tissues. So there's 3 different ways that you exchange through your capillary. So again, you have different types of materials going in and out. So you can go through the endothelial cell cytoplasm using a secretory vesicle, intercellular clefts, so in between your cells, and then lastly, filtration pores. So these are in those fenestration capillary beds where you have more of a need of exchanging through those exchanging from the blood supply and tissue. So there's 4 different mechanisms of capillary exchange. We have diffusion, transcytosis, filtration, and reabsorption. So the first one, we have diffusion. So this is our most important one. So remembering from chapter 1 or chapter 2, one of them. Anyway, diffusion is where stuff is going to go down its concentration gradient. So nutrients like glucose and oxygen are going to go down their concentration gradient out the blood and into the tissue, while waste products like carbon dioxide and your nitrogenous waste, they're going to go down their concentration gradient out the tissues and into the blood. So you can have diffusion, but only if the solutes can actually pass through the plasma membrane, or they're small enough where you can actually get through tiny little holes in between the cells or actually in the cells. So this focuses on only your smaller molecules. So again, this is why your larger blood vessels aren't able to just pop out the capillaries unless they're damaged. Okay, lipids. So your lipids, because of the plasma membrane bilayer, phospholipid bilayer, that's what I'm thinking of. Because that lipid middle section, lipids are actually able to just diffuse across, so just go through your actual plasma membrane. Same thing with oxygen and CO2 that can just pass through the plasma membrane. Water-soluble stuff, they have to find a gap. So either through a pore in the cell, so just an opening in the cell, or those intercellular clefts, so gaps in between your cells. Anything such as your large particles like your proteins, like your albumin, or your big chunky blood cells, they're stuck inside your blood vessels. The last one is transcytosis. So this is kind of a cool one where you have something that's packaged into a vesicle, and that little vesicle just gets transported across your cell membrane and then just emptied out on the other side. Okay, so these next few slides are very important for the Vessels lab exam. Again, your AP2 lab exams are more, the other ones did too, but these are more based on essentially what the same thing you're learning in class. Okay, so filtration and reabsorption. So with our exchange of fluid from our capillaries into our tissue space and back, that's based off of two different types of pressure. So there are going to be pressures inside the capillaries that are working, got pressures in the tissues that are working, and they vary between your arterial end and your venous end. So again, this is how we're going to do our calculations. We actually can show with those measurements the differences with our competing pressures, and you end up with more pressure pushing fluid out the capillary at your arterial end, getting your nutrients to the cell. And then on your venous end, you have more of a pressure difference. Basically, the pressure going into the capillary is larger, and so you end up being able to reabsorb. So on our arterial end, we have our NFP, or our net filtration pressure, NRP, net reabsorption. So filtering fluid out to get those nutrients there, filtering, reabsorbing fluid from the tissues and into your capillaries. So we have two different types of pressure that are working. We have our hydrostatic pressure and our colloid osmotic pressure. So the hydrostatic pressure is very simple. It's just the pressure that the fluid is pushing up against the surface, and that surface in this case is the blood vessel capillary wall. For a colloid osmotic pressure, so remember from chapter 2 we had our different mixtures, and colloid was one of them. So the special ingredient in a colloid being proteins. So focusing on particularly the proteins inside blood do a very big job of this drive. And the interstitial space, so the fluid that is in between the cells, also do have some protein, so they're also going to have a little bit of a colloid osmotic pressure draw, but the one for your blood plasma is a lot stronger, and so you're going to, stronger on your venous end. That helps you to pull fluid in, versus your hydrostatic, your blood pressure on your arterial end is going to be the larger one to cancel out stuff and push fluid out. So both your fluids are going to have both of these pressures. So that's why we have to know where to put those numbers and then do our little calculations. So our net pressures is using both of the pressure from the tissue space and your capillaries for both hydrostatic and osmotic. So for your net hydrostatic pressure, so we're just going to focus on that one portion on one end. So your net hydrostatic pressure on, let's say hypothetically, your arterial end is your blood pressure. So your hydrostatic pressure of the blood is going to push fluid out the capillary. Your hydrostatic pressure of the interstitial fluid, so in between the cells of your tissue, because that pressure is so low, it actually has a negative value. Now, when you do your calculations, ignore the negative value. You just look at it, hey, this is a reminder. This is also helping with drawing fluid out of the capillary. So from our arterial end to our venous end, there is a drop in blood pressure. So there is a drop, or a decrease in the net hydrostatic pressure from the arterial end to the venous end. So you need to know that for the vessel exam. So your blood pressure is higher on your arterial end. Okay, so the other type of pressure is your net osmotic pressure. So this again has to do with osmosis, with the draw of water based off of protein levels. So both your tissue space and your capillaries have proteins. It's just that your your capillary proteins just happen to be a larger amount, and so therefore they have a bigger draw. Okay, so our net osmotic pressure, competing forces between our colloid osmotic pressure of the blood, so that's the fluid being drawn into a capillary. The colloid osmotic pressure of the interstitial space will draw fluid out. But again, they're competing against each other, but the one in the blood, there's more proteins there, so there's more of a pull there. So that's how we're able to draw stuff in on the venous end. So with this pressure, it actually stays pretty roughly similar between your arterial end and your venous end. So it's just that at your arterial end, the blood pressure is large enough to push fluid out of the capillary. So when our capillaries are picking up fluid, it only picks up about 85% of it. The rest of it is with our lymphatic system. So when we go to our next chapter, that's actually what we're covering, the lymphatic system. So there's actually fluid drawn from the tissues into these green-looking lymphatic vessels. And that's also one of the ways, and they have larger spaces to pull stuff in. So that's how some of those virus, bacteria stuff can get into our lymphatic system and help with fighting off that infection, because there's white blood cells that are just hanging out there waiting for them. So the rest of that should be picked up by the lymphatic system. If not, you end up with swelling in the tissues. But we'll get into that more so with chapter 21. So here is an example of our capillary dynamics, looking at our different pressures. So we have the pressures at the arterial end, the pressures at the venous end. So we're looking out here where they have done the calculation. So our net filtration pressure is 33, and this is millimeters of mercury, of pressure when we did our hydrostatic pressure. So that's this calculation here. So the blood hydrostatic pressure, 30 out on the arterial end, but only 10 out on the venous end. So when they did the calculations, you see here you have the bigger arrow is pushing fluid out, so that's going to get nutrients to your cells. On our reabsorption end, you have a bigger arrow of fluid of dealing with your pressures, fluid coming in. So when here they have your net pressures, they also will show you the math down here. So our, and a back. Okay. So our differences here between the 33 out and 20 in. So if they're going in different directions, they're fighting each other. This is where you subtract. If they're going in the same direction, that's where you add. So that's one thing to remember for the vessels exam. So here that difference is 13 out. So pushing fluid out of your capillary. On this side, we have 13 out versus 20 in. Again, opposite direction, you subtract. So you end up with a difference of 7, so bringing 7 in. So for that one, that is your reabsorption of the fluids from your tissue space into your capillaries. Okay, so here's just kind of a more basic version of that. So capillary filtration at the arterial end, nutrients to the cell, capillary reabsorption at your venous end, bringing waste from the cells. Okay, so done with the vessel exam stuff, and anything else that's necessary or needed for that exam will have a star on that slide. Okay, so with your capillaries, usually they absorb most of it. And again, your lymphatics do some of that work. But there are some exceptions. So for example, your kidney capillaries do not reabsorb. That fluid that gets filtered out of your capillaries stays in the kidneys because it's going to get processed into either urine or stuff that get taken back up from the kidney tubule structures, and then that gets re-added back to your blood. So for example, there's some smaller molecules that's going to get filtered out in the kidneys, like glucose, but your body takes back all of that glucose if you're healthy. Now, if you have like diabetes and you have high blood sugar, then you run out of channels to bring glucose from the filtered out fluid in your kidneys and send it back to your blood. So that's one of the tests that they do for diabetics, or see if someone is like pre-diabetic, is they test their urine. If there's glucose in the urine, then that could be a signal that their blood sugar is too high. Okay. The alveolar capillaries, they're going to completely absorb fluid, and that's just because you don't want fluid in your lungs because you can't breathe. Okay. Depending on your activity, it's going to affect what's going on in your capillaries. So at rest, you have more of a reabsorption because your blood pressure is low. So you don't have that active push from your heart moving fluid to your tissues because you're doing stuff. Since you're sitting on your butt, you don't need all that. So you have more reabsorption, which helps to clear out the stuff out of your tissues. Anything that's active is going to have an increase in capillary flow and blood pressure because you are moving, you're doing stuff. You need to get the nutrients to those tissues so they can do what they need. So that's why if you're ever looking like backstage at a muscle builders convention where you'll see them using like resistant bands, or they're doing push-ups, or they're doing whatever, even though they're like dehydrated and haven't eaten a while, but because that's what gives them that kind of shrink wrap look. What they're doing is they're trying to get that blood to pump up their muscles. It's kind of like your last time to kind of get that pump right before you get on stage, and that shows off your muscles even more. Okay, so let's say if your capillaries don't pick up the fluid like they should, you end up with what's called edema, fancy word for tissue swelling. So there's excess fluid in your tissue. So 3 causes of that. Increased capillary filtration. So this could be caused by kidney failure, so your kidneys aren't doing their job, so you end up with a bunch of excess fluid that gets pushed out because you're not making urine. Excess fluid gets pushed out to your tissues. A histamine release. So that's part of your immune response. So that's why a antihistamine is what you do to fight off whatever type of allergy that you have. So that's why, for example, for some people in their nasal passages, they get all clogged up because that histamine from the immune response of that pollen or dust or whatever they're allergic to causes swelling in those tissues because they, for example, breathe that stuff in. Old age and poor venous return. So that's just kind of, you just have banged up blood vessels. And so if you're not as healthy, not as active, then you end up possibly having tissue swelling. Next one, reduced capillary absorption. So this is not picking up that excess fluid. So this could be low protein levels. So that means you have a lower colloid osmotic pressure of the blood. So less pressure pulling fluid into your tissue, into your capillaries. There's some form of liver damage, which could also lead to your hypoproteinemia. So your livers aren't, not livers, liver isn't making the albumin protein like it should. So low protein levels, or just not enough protein in your diet, so you don't have the amino acids to make the necessary proteins. And then lastly, obstructed lymphatic drainage. So your lymphatic system, that picks up the last bits of that last 15% of fluid that gets dropped off. If those aren't picking up fluid like they should, then what can happen is that they remove your lymph nodes. So that's sections that help with filtering out that lymph And so that would slow down, even though they're good for your immune system to function, that's also slowing down the return of the fluid from the tissues and back to your circulatory system. Okay, consequences. So if all this fluid is going into your tissue, it interrupts your nutrient and waste exchange, so it could cause your tissues to die off, or necrosis, which is just a fancy word for death. If it happens in the lungs, you have pulmonary edema, so build up in the lungs, so that can cause you to be to suffocate. So that's why some of these people now with COVID, and they end up with pneumonia, they end up having fluid build up in the lungs and therefore can't breathe. Next one, cerebral edema, so happening in your brain, so that causes increased pressure with headaches, nausea, just because it interrupts your balance systems, as well as seizures and a coma, just because you are affecting your brain activity because you just have all this excess pressure. Severe edema, so excess fluid causing low blood volume because that fluid isn't returning back to your blood, so that decreases how much you have, so that's less ability of your blood to circulate your nutrients. And low blood pressure as well, so that the fluid isn't in those arteries, and so you don't have that push to be able to move fluid into your tissue space. So circulatory shock is just where you've lost too much blood. Okay. So we are going to start here for the next video.